I really appreciate this kind invitation to share with you some data about obesity and cardiovascular protection. And it's a pleasure to be here in the webinar week of metabolism and target organ damage. As you heard, I'm a professor of medicine, a professor of physiology at the Mayo Clinic, Division of Nephrology and Hypertension. And why are we concerned about obesity? Well, you may know that the prevalence of obesity is continuously on the rise. And as you can see, it includes many populated continents, such as Africa, the Americas, as well as Asia. This is a very alarming rise in prevalence because we know that obesity can cause various forms of cardiovascular and other forms of damage, including aggravating diabetes, cancer, cardiovascular, and kidney disease. In addition, that obesity can also increase susceptibility to other forms of injury, serving as a second hit. The underlying mechanisms for this increased susceptibility to damage have not been fully clarified, but it has been postulated that obesity interferes with normal defense and repair processes. When we talk about the bodily defense system, we talk about specifically cellular defense systems, which can be composed of both circulating cells as well as tissue resident cells. We know that when regions in the body are injured, they release regulatory chemokines and cytokines that mediate the mobilization, homing, and transdifferentiation of blood marrow-borne endothelial progenitor cells that are also called EPC. Those EPCs can promote vascularization as well as cellular replacement of injured endothelial cells. In addition to the circulating system, there's also a tissue resident repair system. Most of the tissues contain niches of local resident stem cells that respond to local cues and subsequently participate in the repair of neighboring cells, primarily by the release of paracrine factors, such as anti-inflammatory factors and growth factors that can decrease inflammation and induce angiogenesis. However, those cells themselves are not exactly invulnerable to disease. Under normal conditions, the stem cells reside in a stem cell niche under quiescent conditions. However, obesity can disrupt the stem cell dynamics. Altered metabolic signaling in the stem cell niche after exposure to various inflammatory factors, high glucose levels, reactive oxygen species, advanced glycation end products, and others, can lead to loss of stem cell quiescence and activation of differentiation, preferentially into adipocytes. This means that this pool of stem cells is exhausted and they are no longer available to recruitment upon need. There is also increased presence of inflammatory cells in this niche within obesity. What are the mechanisms that are responsible for the impairment of reparative cells in obesity? Research studies have primarily implicated inflammation in this process. It has been shown that obesity induces activation of Th1 lymphocytes, as well as polarization of macrophages towards the pro-inflammatory N1 phenotype. This means that it induces adipogenesis, decreases osteogenesis and proliferation, and also increases cellular senescence, which may lead to premature cellular death. All of these interfere with stem cell function. What mechanism can make this happen in obesity? This slide shows some recent preliminary MEDIPSIC 5-HMC studies in which we compared epigenetic modifications in adipose tissue-derived mesenchymal stem cells, or MSCs, that were obtained with human subjects with obesity compared to lean human subjects. On the left, you can see a heat map that shows average 5-HMC levels in human lean and obese MSCs. This one is in 3 samples each. You can see in red that obesity induces activation of genes that are associated with cellular senescence, inflammation, and epigenetic factors. And on the other hand, downregulates the activity of genes that are associated with angiogenesis, as you can see here in blue. On the right panel, you can see the corresponding integrative genomics viewer browsers or IGV maps of obese, green, and lean in cyan genes. We found that there are indeed higher 5-HMC levels in regulatory regions of senescence genes, inflammatory genes, and epigenetic factors, suggesting that obesity induces epigenetic changes in the activation of genes in adipose tissue-derived mesenchymal stem cells. In addition to these epigenetic changes, obesity may also induce changes in the expression levels of mRNAs and microRNAs in several important pathways. Normally, healthy adipose tissue-derived MSC express genes that are associated with anti-inflammation, reduced oxidative stress, and decreased fibrosis, for example, in the tubulointerstitial area of the kidney and other regions as well. On the other hand, in obesity, there is increased activation and expression of genes that are associated with inflammation, apoptosis, interruption of mitochondrial function, and senescence. As a result, the endogenous repair systems may be impaired. And this means that it has implications not only for endogenous repair, but also if you want to use those cells for cell-based therapy, the use of autologous cells might be unadvisable because they may be damaged. In particular, we found over the years that obesity induces a specific impairment in the mitochondrial structure and function in mesenchymal stem cells. For example, in the left panel, you can see in the top images of transmission electron microscopy that show mitochondria from lean MSC compared to obese MSC. You can see that obese MSC show increased swelling and loss of cristae structure. Here you can see staining for a mitosox, which detects mitochondrial oxidative stress, which is clearly increased in obese MSC, as you can see in red. On the other hand, TMRE staining that detects the mitochondrial membrane potential is significantly decreased in obese MSC. This is associated with a significant change in gene expression of genes that are encoded by the mitochondrial circular DNA, such as ND2, ND4, and others. Interestingly, we found that obesity also upregulates the expression of a specific mitochondrial target microRNA called mitomer, or mitochondrial microRNA 181A, in human mesenchymal stem cells. This is a specific mitochondrial mRNA that can be imported into the mitochondria. This is why we ascribe important function for it. And you can see that it is increased in expression in my obese MSCs, both in the IGV as well as qPCR, suggesting that it might be involved in the effects of obesity on MSCs. Zhang Lei is now working on this important manuscript. What is the consequence of this? We know that the mitomeres can affect the expression of mitochondrial-derived peptides, or MDPs, such as MOTSI. MOTSI, mitochondrial open read frame of the 12S RNA, has an important role in regulation of metabolism, and its levels decline in aging and early cardiovascular disease. Interestingly, we found that MOTSI is downregulated in obese MSC. You can see here, this is again the circular DNA of the mitochondria. You can see the decreased expression of the 12S and D-loop MDPs, and this was specifically in MOTSI, whose level were decreased. Si-Hyun is now in charge of this project. As a result of these changes in mitochondrial function and gene expression and activation, obesity can impair the reparative function of adipose tissue-derived MSC, both in vitro and in vivo. For example, this study shows some studies that we performed in vitro, and you can see here the comparison of proliferation of lean versus obese MSC. You can see the proliferation of obese MSC is reduced compared to lean. This is associated with upregulation of the prosenescence mediator P16 in obese MSC, as well as in upregulation of interleukin 6, an important inflammatory factor. In the bottom, you can see that this is also associated with impairment of reparative function as they affect other cells. Here we incubated MSCs with UVEX. Normally, UVEX induce vascular formation or tube formation in the matrigel assay, but when the UVEX are injured, there is a decrease in the function and angiogenesis potential of those cells. When we incubate them with lean or non-obese MSCs, they restore the angiogenic function of the UVEX, whereas obese MSC failed to induce an increase in the number of tubes, suggesting that the obese MSCs interfere with the angiogenic function of injured UVEX and lost their reparative function. This also translates into impaired in vivo function. We performed a study in which we infused MSCs into 129 mice that were either healthy, sham, or had renovascular hypertension. We induced renovascular hypertension by putting a little clamp or a tube on the renal artery, one renal artery of these mice. And this induces a cardiovascular injury, it induces hypertrophy of the heart and other forms of injury. In these mice, we infused either lean MSCs or obese MSCs. You can see here the MSCs were detected in the myocardium, suggesting that indeed they reach the myocardium because we infused them into the carotid artery. In several studies, we tested the function of those MSCs on the heart. We found that here in the left, you can see a WGA staining, with agglutinin staining of the heart, in which we test and study the cross-sectional area of those cardiomyocytes. We found that indeed in renovascular hypertension, there was an increase in the cross-sectional area of the myocytes, suggesting hypertrophy, which was significantly attenuated, although not normalized, by lean MSCs, but not by obese MSCs. Interestingly enough, even in sham animals, infusion of obese MSCs induced some hypertrophy of the myocytes, suggesting that obese MSCs alone can increase cardiovascular injury. On the bottom panel, you can see cardiac fibrosis that was detected by trichome staining. And again, you can see increased fibrosis in renovascular hypertensive hearts. However, they were similarly reduced by both lean MSC and obese MSC, suggesting that not all the function of obese MSCs are impaired. In addition, we also studied the effects of the MSCs on oxidative stress in capillary count in RVH hearts. On the left panel and in the right panel, you can see there is increased oxidative stress in the hypertensive heart, as detected by 8-OHDG staining. And this was attenuated by lean MSC, but much less so by obese MSCs. And again, like we observed for hypertrophy, we find that infusion of obese MSCs alone can increase oxidative stress in the hearts exposed to renovascular hypertension. On the other hand, you can see on the bottom panel staining for capillaries and capillary count by CD31 staining, and we can see that there was a significant loss of capillaries or capillary rarefaction in the hypertensive heart that was rescued by lean MSCs as well as by obese MSCs, suggesting that they didn't lose all the reparative function. These observations about the mechanisms by which MSCs can be impaired in obesity led to the discovery of several potential therapeutic targets. For example, I showed you some slides showing the decrease in the expression of MOTSI and increase in the expression of mitomer 181A in obese MSCs. Therefore, we performed a couple of studies, both in vitro and in vitro, to test the ability of targeted interventions to improve the function of those MSCs. On the right panel, you see a study in which we co-incubated MSCs with a anti-mitomer 181A. This is an antagonist antagomer that blocks the function of mitomer 181A. You can see again TEM images on the top showing again the swelling of the obese MSC and the loss of cristae structure. And you can see that even visually, treatment with obese MSC that have been pre-incubated with anti-MIR 181A restore the structure of these obese MSCs. In the middle panel, you can see again the staging for mitosocs that indicates oxidative stress in the mitochondria. You can see again the increased oxidative stress in the mitochondria of obese MSCs that was blocked by treatment with anti-MIR 181A. Similarly, the anti-MIR 181A also restored membrane potential of the mitochondrial as indicated by mitomer, by staining for TMRE. On the left, you can see some in vivo studies in which we infuse MSC that had been pre-treated with MOTSI. This is the mitochondrial-derived peptide that I mentioned a little earlier. We used a similar protocol to the one I showed you before. And you can see here on the top panel DHE, staining dihydroerithidium, which detects cardiac oxidative stress. We can see that in RVH, or the hypertensive heart, there was a significant increase in oxidative stress. And this was attenuated by lean MSC, but not by obese MSC. However, pre-incubation with MOTSI that restored the level of MOTSI in the obese MSC improved the reparative function of these obese MSC because it allowed them to decrease oxidative stress in the hypertensive heart. On the bottom panel, you see CD31 staining, which details capillary count in a hypertensive heart. And you can see again that there is significant increase in the capillary count in the heart of those hypertensive mice. This was not significantly improved actually by pre-treatment with MOTSI, suggesting that perhaps MOTSI does not regulate the angiogenic function of the MSCs. Nevertheless, treatment with MOTSI allowed the MSCs to improve the capillary count, suggesting that might be some other pathway involved by which MOTSI improved regulation of angiogenesis and increased the count of capillaries. These are not the only interventions that improve mitochondrial health in newborn obese MSC. In this slide, you see a study in which we studied obese MSCs before and 6 to 9 months after bariatric surgery that induced a significant weight loss in the individuals. You can see here that the area of mitochondria was significantly increased in obese MSCs due to swelling, as I mentioned earlier. But bariatric surgery induces significant reduction in the area of the obese MSC, suggesting improved decreased swelling. And you can see it also here in the images that the mitochondria resume their elongated and less swollen appearance. In addition, we also measured matrix density. This measures the structure of the cristae. You can see here a significant decrease in the density of the mitochondria compared to lean mitochondria. And bariatric surgery resumed the structure and the function of these mitochondria. So we can summarize this finding by saying that obesity appears to impair the function as well as the structure of mesenchymal stem cells, MSCs, both in vivo and in vitro. This involves a significant mitochondrial dysfunction. It involves cellular senescence, and we know that cellular senescence, as I mentioned, can induce premature death, but also senescent cells can release inflammatory factors. This is called SASP, and affect also neighboring cells. So overall, this impairs the reparative capacity of mesenchymal stem cells. The mechanisms by which obesity impairs the function and structure of mesenchymal stem cells may involve epigenetic modifications, as well as changes in the expression and function of mitochondria-related microRNAs. So to conclude, obesity may increase susceptibility to injury by interfering with normal defense and repair processes associated with MSCs. And I haven't shown you here because for the sake of time, but they also impair the number and function of endothelial progenitor cells. This may have implications for endogenous repair processes because these cells repair endogenous systems such as parenchymal cells as well as vascular cells. And this may also affect the ability of using both MSCs and EPCs for exogenous repair when we want to use them for cell-based therapy to regress and reverse some of the effects of obesity or other diseases. This involves possible epigenetic dysregulation that can also extend not only to mesenchymal stem cells, but also to other immune and structural cells, like parenchymal cells of various types. And we believe that elucidation of the mechanisms that are responsible for the impaired cellular repair in obesity may help direct developments of targeted therapeutic interventions, as I also showed you, that can improve their function both in situ as well as in vitro. I also want to take the opportunity to thank the members of my lab. And you can see here Zhang Lei, for example, who have done some very important and critical studies, as well as other members, both from China and other countries, as well as the United States. Thank you very much for your attention, and I will be very happy to take questions.